Hello everyone. Today let's discuss the Unit 3 Cell Structure and Functions. Biology is the study of living organisms. The detailed description of their form and appearance only brought out their diversity. It is the cell theory that emphasized the unity underlying their diversity of forms, that is, the cellular organization of all life forms. A description of cell structure and cell growth by division is given in the chapters comprising this unit. Cell theory also created a sense of mystery around living phenomena, that is, physiological and behavioral processes. This mystery was the requirement of integrity of cellular organization of living phenomena to be demonstrated or observed. In studying and understanding the physiological and behavioral processes, one can take a psychochemical approach and use cell-free systems to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by analyzing of living tissues for elements and compounds. It will give us what type of organic compound are present in the living organism. In the next stage, one can ask the question, what are these compounds doing inside a cell? And in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is the molecular basis of all physiological processes? It can also explain the abnormal processes that occur during any diseased condition. The psychochemical approach to study and understand living organism is called reductionist biology. The concept and technique of physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules is provided. G.N. Ramachandran. An outstanding figure in the field of protein structure was the founder of the Madras School of Conformational Analysis of Biopolymers. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformations of protein through the use of the Ramachandran plot rank among the most outstanding contributions in structural biology. He was born on October 8, 1922 in a small town not far from Cochin on the southwest coast of India. His father was a professor of mathematics at the local college and thus had considerable influence in shaping Ramachandran's interest in mathematics. After completing his school years, Ramachandran graduated in 1942 as the top ranking student in the BSc Honour Physics course of the University of Madras. He received a PhD from Cambridge University in 1949. While at Cambridge, Ramachandran met Linus Pauling and was deeply influenced by his publications on models of the alpha helix and beta sheet structure that directed his attention to solving the structure of collagen. He passed away at the age of 78 on April 7, 2001. Chapter 8 Cell, the Unit of Life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of the basic unit of life, cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence, and 2. performing the essential function of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anthony von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural detail of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissue of the plant. At about the same time, Theodor Schwann, 1839, a German zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is the unique character of the plant cell. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleiden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divide and new cells are formed from the pre-existing cells, omnicellular, acellular. He modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Cell theory as understood today is, first point, all living organisms are composed of cells and products of cells. And the second point, all cells arise from pre-existing cells. An overview of cell. You have earlier observed cell in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called a nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material, DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cell, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Beside the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelle, like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosome are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria, and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. Cells differ greatly in shape, size, and activities. For example, mycoplasmas, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. So let's look on the figure 8.1. The first figure shows red blood cells, which is biconcave and round shaped. The second figure shows the picture of white blood cell, which is amoeboidic. Then the third figure is columnar epithelial cells, which can be seen as long and narrow one. The fourth one is the nerve cell, which is the longest cell, which is branched and very long. Okay. A trachea, elongated cell, and mesophyll cells, round and oval. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cell. We can see that in the figure 8.2. A typical eukaryotic cell 10 to 20 micrometer. Typical bacteria 1 to 2 micrometer. PPLO, which is pleuropneumonia-like organism, which is about 0.1 micrometer. And viruses, which is 0.02 to 0.2 micrometer. This is the diagram showing comparison of eukaryotic cell with other organisms. They may vary greatly in shape and size. The four basic shape of the bacteria are Bacillus rod-like, Coccus spherical, Vibrio coma-shaped, and Spirillum spiral-shaped. The organism of the prokaryotic cell is fundamentally similar even though prokaryotes exhibits a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The semi-fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, a single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic character to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. The nuclear membrane is found in eukaryotes. No organelles like the one in the eukaryotes are found in the prokaryotic cell except for ribosomes. The prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfoldings of the membrane. The cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram. Those that take up the Gram stain are Gram positive and the other that do not are called the Gram negative bacteria. The glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamella. They help in the cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells, which may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria shows a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are also known to help attach the bacterium to rock in streams and also to the host tissues. Ribosome and inclusion bodies. In prokaryotes, ribosomes are associated with plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S unit, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA into protein. Inclusion bodies. The reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. They are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophysian granules, glycogen granules. Gas vacuoles are also found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. The eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition to eukaryotic cells, have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell wall, plastids, and a large central vacuole, which are absent in animal cell. On the other hand, the animal cells have centrioles, which are absent in almost all plant cells. Let's see the figure 8.3. Here we can see the picture of a typical plant cell. So this is the nucleus. We can see that this is the nucleus and this portion is the nucleolus. And this portion is called the Golgi apparatus. And here we can see the nucleus is covered by a nuclear envelope. Then very close to the nucleus here we can see the rough endoplasmic reticulum and also closer to this smooth endoplasmic reticulum. And here we can see, and other than rough endoplasmic reticulum, here we can see the Golgi apparatus. Then here we can see the lysosomes and here we can see the large vacuole. Okay, and this is the plasma membrane of the cell. The central portion is the middle lamella, and outer portion here we can see the cell wall. Okay, this is mitochondria, and these small structures are called the ribosomes, and this is the chloroplast. And here we can see in between all these cell organelles we can see the cytoplasm, and this small structure is called perioxisome. Here we can see the perioxisome, and this is the microtubule. Okay, and these small junction, here we can see that these small junctions are called a plasmodesmata. So we finished that portion. Let's move on to animal cell. Here we can firstly see the microvilli, small projections. Then we can see the plasma membrane, which is an outer delimiting structure of an animal cell. Then we can see the nucleus, and here we can see the nuclear envelope, the portion covering the nucleus. And here we can see inside the nucleus a structure called the nucleolus. Then adjacent to the nucleus, here we can see the rough endoplasmic reticulum, and in adjacent to that, here we can see the smooth endoplasmic reticulum. Here we can see the Golgi apparatus, like this Golgi apparatus. Then we can see the lysosome here, and usually an animal cell doesn't have a vacuole. And we have to say centriole. Here we can see the centriole. Then perioxisome can also be seen here, perioxium. And let's see, this is the mitochondria in the animal cell, and here we can see in between the fluid matrix, which is seen in between the organelle, is the cytoplasm. And okay, we completed all those portions. Let us now look at individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, the chemical studies on the cell membrane, especially in human red blood cell, enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outside and the hydrophobic tails towards the inner part. Once again, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails toward the inner part. This ensures that the nonpolar tail of the saturated hydrocarbons is protected from the aqueous environment. Here we can see in the figure 8.4. Okay. This is the phospholipid bilayer. Here we can see that. We can see two types of protein, peripheral and integral protein here. We can see sugar. Okay. Then, in addition to the phospholipids, membrane also contains cholesterol. Later biochemical investigations clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipids. Depending on the ease of extraction, the membrane protein can be classified as integral and peripheral. Peripheral protein lie on the surface of the membrane, while the integral protein are partially or totally buried in the membrane. Here we can see that the fluid mosaic model of plasma membrane. It is called as the fluid mosaic model. Please note that name, fluid mosaic model of plasma membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model. Figure 8.4. According to this, the quasi-fluid nature of lipid enables lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of function like cell growth, formation of intercellular junctions, secretions, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecule present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. The neutral solute may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower concentration. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot move through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are passed across the membrane against their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example, Na plus or K plus pump, sodium or potassium pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. The cell wall not only gives shape to the cell and protects the cell from the mechanical damage and infection, it also helps in the cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and protein. The cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner towards membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and the middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of the membranous organelles is distinct in terms of structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum (ER), Golgi complex, lysosome, and vacuole. Since the function of the mitochondria, chloroplast, and perioxysomes are not coordinated with the above components, these are not considered as part of endomembrane system. The endoplasmic reticulum. Electron microscope studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum ER. Here we can see that it is in the figure 8.5. This is the nucleus, and here we can see the nuclear pore and rough endoplasmic reticulum closer to the nucleus, and can be seen the ribosomes. Smooth endoplasmic reticulum can also be seen in this picture. Hence, Endoplasmic reticulum divides the intracellular spaces, not the point, intracellular spaces into two distinct compartments, that is luminal inside endoplasmic reticulum and extraluminal cytoplasm compartments. The endoplasmic reticulum often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called a rough endoplasmic reticulum, RER. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum, SER. Here we can see that clearly in this picture. We can see the rough endoplasmic reticulum. On the surface of the endoplasmic reticulum, here we can see the ribosomes. Hence it is called the rough endoplasmic reticulum because its surface is much rough compared with the smooth endoplasmic reticulum, which doesn't have any ribosomes on its surface. RER, which means rough endoplasmic reticulum, is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER. The Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of 0.5 micrometer to 1 micrometer diameter. Here we can see that in the figure 8.6. Golgi apparatus, and we can see the cisternae here in the picture. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and the concave trans or the maturing phase. Once again, convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the endoplasmic reticulum, fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. The Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging on the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolases, lipases, proteases, carbohydrases. Optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acid. Here we can see lipases which digest lipids, proteases which digest proteins, carbohydrases which digest carbohydrates, and here we can see nucleic acid, and the nucleic acids are digested by nucleases. Okay. Let's move on to vacuole. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory products, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuole can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against the concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. Mitochondria. Mitochondria, singular mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell, in terms of shape and size also. Considerable degree of variability is observed. Typically, it is a sausage shaped or cylindrical, having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer, and length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane, dividing its lumen distinctly into two aqueous compartments, that is, the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infoldings called cristae, singular crista, towards the matrix, which can be seen in the figure 8.7. The cristae increase the surface area. The two membranes have their own specific enzyme associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses single nuclear DNA molecule. A few DNA molecules, ribosomes of 70S type, and the components required for the synthesis of protein. The mitochondria divide by fission. Here we can see the figure 8.7, the structure of mitochondria by the longitudinal section. Here we can see the outer membrane, and inside the outer membrane we can see inner membrane, and in between them we can see intermembrane space. We can see the infoldings of the mitochondria of the inner membrane, infoldings of the inner membrane crista, and can also see in the matrix portion. Plastids. Plastids are found in all plant cells and in the euglenoids. They are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contains chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigment like carotene, xanthophyll, and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastids of varied shape and sizes with stored nutrients. Amyloplast stores carbohydrates, starch, example potato. Elioplast store oil and fat, whereas the eleuroplast stores protein. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval-shaped, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the chlamydomonas, a green algae, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. The number of organized flattened membranous sacs called the thylakoids are present in the stroma, figure 8.8. Thylakoids are arranged in stack like the piles of coins called grana, singular granum, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamella connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small, double stranded, circular DNA molecules and ribosomes. The chlorophyll pigment are present in the thylakoids. The ribosome of the chloroplast are smaller 70S than the cytoplasmic ribosome of 80S. Here we can see the detailed view of the sectional view of the chloroplast. Outer membrane, intermembrane, granum, thylakoid and stroma lamella. Ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Palade in 1953. They are composed of ribonucleic acids (RNA) and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunits, which can be seen in the figure 8.9. Here we can see smaller subunit and large subunit. Large subunit is in almost violet color and small subunit which can be seen as in pink or rose color. The two subunits of 80S ribosomes are 60S and 40S while the 70S ribosomes are 50S and 30S. Here S Swedberg unit stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Once again repeating that 80S ribosomes are composed of two subunits, 60S large and 40S small, and 70S ribosomes are composed of two subunits which is of larger is 50S and smaller is 30S. Cytoskeleton. An elaborate network of filamentous proteinaceous structure consisting of microtubules, microfilaments and intermediate filaments present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Here we can see in the figure 8.10 the section of cilia or flagella showing different parts. The figure A represents the electron micrograph view of cilia and flagella, and the detailed view can be seen in the diagram B, which represents the internal structure. Here we can see the plasma membrane. Here we can see the plasma membrane here. And these are the peripheral microtubules which can be seen as doublets. And this is the interdoublet bridge. Here we can see the interdoublet bridge in between the doublets. And this is the central microtubule. This is the central microtubule. Central microtubules are of two. Here we can see the radial spoke connecting to the central sheath from each doublet. And this portion is the central sheath portion. Okay. Cilia and flagella. Cilia, singular cilium, and flagella, singular flagellum, are hair-like outgrowth of the cell membrane. Cilia are small structure which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a cilium or the flagellum shows that they are covered with plasma membrane. Their core, called the axoneme, possess a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of the axoneme microtubule is referred to as 9 plus 2 array, which can be seen in the figure 8.1, which we discussed earlier. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. Centrosomes and centrioles. Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplet are also linked, and the central part of the proximal region of the centriole is the proteinaceous and called the hub, which is connected to the tubule of the peripheral triplet by radial spokes made of protein. The centriole form the basal body of cilia or flagella and spindle fiber that give rise to spindle apparatus during the cell division in the animal cell. Nucleus. Nucleus as a central organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dye was given the name chromatin by Fleming. The interface nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin. Nuclear matrix, one or more spherical bodies called nucleoli, singular nucleolus, which we can see in the figure 8.11. is the structure of nucleus, can be seen the nucleoplasm, and nucleolus can be seen, which is like a solid structure, and also here we can see the nuclear membrane. Electron microscopy has revealed that the nuclear envelope, which consists of 2 parallel membranes with a space between 10 to 50 nanometer, are the perinuclear space, form a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its 2 membranes. These nuclear pores are the passages through which movement of RNA and DNA molecule takes place in both directions between the nucleus and the cytoplasm. Normally, there is only 1 nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than 1 nucleus per cell? Some mature cells even lack nucleus. Example, the erythrocytes of many mammals and the sieve tube cells of the vascular plants. Would you consider these cells as living? The nuclear matrix of the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and distinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic protein called histones, some non-histone proteins and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 or 23 pairs chromosomes. You will study the details of DNA packaging in the form of chromosome in class +2. Every chromosome, visible only in dividing cells, essentially has a primary construction or the centromere, on the sides of which disc-shaped structures called kinetochores are present, which can be seen in the figure 8.12. The centromere holds two chromatids. The centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosomes can be classified into 4 types, which can be seen in the figure 8.13. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In the case of acrocentric chromosome, the centromere is situated close to its end, forming the extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Here we can see the detailed structure of the type of chromosomes based on the position of the centromere. The first one is the metacentric chromosome. The second one is the submetacentric chromosome. Third one is the agrocentric chromosome, and the last one is the telocentric chromosome. And here we can see the centromere of each chromosome, and here we can compare the length of the arms or the chromatids upper and below the centromere. Sometimes a few chromosomes have non-staining secondary construction at a constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plants and animals. Let's move on to the summary portion. All organisms are made up of cells or aggregates of cells. Cells vary in their shape, size, and activities or functions based on the presence or absence of membrane-bound nucleus and other organelles. Cells or hence organisms can be named as eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus, and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transport of several molecules. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. All cell organelles perform different but specific functions. The centrosome and the centriole form the basal body of cilia and flagella that facilitate locomotion. In animal cells, centrioles also form spindle apparatus during cell division. The nucleus contains nucleoli and chromatin network. It not only controls the activities of the organelles but also plays an important or major role in heredity. The endoplasmic reticulum contains tubules or cisternae. They are of two types, rough and smooth. Endoplasmic reticulum helps in the transport of substances, synthesis of protein, lipoproteins, and glycogen. The Golgi complex is a membranous organelle composed of flattened sacs. The secretions of the cells are packed in them and transported from the cell. The lysosome are single membrane structures containing enzymes for digestion of all type of macromolecules. The ribosomes are involved in protein synthesis. These occur freely in the cytoplasm or are associated with endoplasmic reticulum. The mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate. They are bound by double membrane and outer membrane is smooth and inner one folds into several cristae. The plastids are pigments containing organelles found in the plant cells only. In plant cells, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reactions and the stroma of dark reactions. The green colored plastids are chloroplasts which contain chlorophyll, whereas the other colored plastids are chromoplasts which may contain pigments like keratin and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membranous structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, the cell is the structural and functional unit of life. So, here comes the end of this chapter. Keep going. Thank you.